Yes, I'm going to be telling you about how bacteria can live on air. So to start with, you may have seen or heard of this TV show Alone. I'm just going to play a clip from it.
This is a whole new level of survival, and we're trying to film it.
I commend anyone who can survive in this area.
There's a bear right across.
I feel like I'm starving.
I didn't come here to get killed.
It's going to be hell.
It's so loud up there. Hey, bear.
So I guess in the first iteration it was only men that were daring enough to do it. But I think now there's women that do it. So essentially the premise is they're like dropped in the middle of nowhere and they have like very few items that they can use to survive, and they have to survive the longest. And the longest wins. And so I think the longest someone survived was 100 days, which is pretty long considering, you know, I don't really like being out in the cold for like 10 minutes. But this is not actually like the best survivalists. I think as humans we think of, you know, this sort of thing as incredible survivalism. But actually the best survivalists that exist are bacteria. And so bacteria, I think, are commonly overlooked in this respect. We often think about them as things that cause disease, like antibiotic resistance, that sort of thing. We think of them as things that can cause food poisoning, or if like you have some chicken you left in the fridge, you're worried you might get salmonella. But actually bacteria are kind of everywhere, and they're doing a lot more than we think. So they're actually not just, you know, disease causing, they can actually be neutral or kind of good bacteria, I guess. And so bacteria are incredibly abundant, and they inhabit a very, very large amount of environments that we don't really think of, that aren't occupied by humans or plants or even animals. And these are like the most extreme environments. So think of like volcanoes or like under like a meter of ice in Antarctica. Bacteria are pretty much everywhere. And the commonality of a lot of these places is soil, and so bacteria are incredibly abundant in the soil. So something like 1 gram of soil is like way less than a teaspoon. There's 40 million bacterial cells. And so this also like serves as a reminder when you're gardening, make sure you wear gloves because they're everywhere. And bacteria are doing a lot of things in soil. And so in the soil, generally, they're eating sugars, which is the same as us. So we eat sugars a bit more complex form. So whatever you have for dinner is obviously made up of sugars. And that's integral for our cells in how they get their energy. Bacteria get sugars in a much easier form, generally from like either like plants decomposing or other things kind of just like leaving sugars in the soil. And they like to consume this preferentially. And they'll consume this as much as they can until potentially they find themselves without sugars. And this could be because they're in really nutrient-poor soils, like in Antarctica, it's very nutrient-poor. Or it could be because there's a lot of the bacteria around, so even if they're in like a rich soil, like the potting mix you get from Bunnings, they could have a lot of bacteria in this. They just don't have enough nutrients because they're all trying to eat the same thing. It could also be because they're like, for instance, like waterlogged, there's extreme weather events. And so basically the flux of this sugar components of the soil is changing all the time, irrespective of where they live. And so they often find themselves starving, but they need to survive. And so bacteria kind of have adapted to survive by entering a state of dormancy. And this is essentially like a really big sleep. And so they basically shut down the metabolism, they go into dormancy because if they don't survive, they die, and then all the bacteria just die, and that's obviously not what we want. And so to survive, they do need tiny, tiny amounts of energy. And so instead of getting it from the soil and from the sugars, they get it from gases in the atmosphere. And so they get it specifically from trace gases. And so they're able to just like exist in the soil, and because gases are everywhere, irrespective of the environment, they're able to just passively absorb these gases and make energy. And so what are trace gases? Just as a reminder in case you don't know, most of the atmosphere is composed of nitrogen, so this is 80%. Then there's oxygen, which is obviously pivotal to our life. And then about 1% remains the trace gases. And so there's a number of trace gases in this 1%, and the one that I look at is hydrogen. And so there's 0.00005% hydrogen in the atmosphere, which is obviously like incredibly tiny amounts. And these bacteria are able to extract that and use it for energy. So like of all the molecules floating around them, they're able to pick out selectively hydrogen and use that to fuel their survival. And so how do they do this? So this is kind of like schematic of a bacterial cell, just one cell. And so bacteria are single-celled, so they obviously need to try and survive as much as possible and be the best survivalists. And so outside the cell, they have this membrane that basically contains everything within it. And then within that membrane, they have all the stuff they need to do the jobs. So they have like DNA, they have proteins. And this protein that I've kind of magnified in on is a specialized enzyme, which is like a molecular machine, that takes hydrogen and makes it into energy for the cell. And so it basically is able to extract hydrogen in the tiny, tiny amounts in the atmosphere and then convert this to electrons. And so electrons, when they're flowing, is electricity. And then the cell harnesses this and basically makes energy and then is able to survive. And so we published this work in quite a big journal called Nature. And the concept of this was that we could extract this enzyme, this little molecular machine, and we're basically able to show in the lab that when you supply them with just the air that we're breathing, so with the tiny amounts of hydrogen in it, they can make a very, very small amount of electricity. And like emphasis on the very small. But it kind of went from like my PhD project one day to when it was released being like this huge media thing that we were generating electricity from the air. And so some weird publications came out about this. So enzyme that can help make energy dreams come true. So I think people were kind of convinced that we were like going to be running watches or something, or potentially cars on our enzyme. But again, very small amounts of electricity. This is a picture of my boss, I think is so funny. The scientists find enzyme that can make electricity out of tiny amounts of hydrogen. I mean, this is true, but you know, it's making tiny amounts of electricity too. And then there's a lot like this where they have this little picture of the enzyme and then like insinuating that it's powering a light. Enzymes generating electricity from air. And then there's this little like movie summarizing what happens. So we have the bacteria in the soil and then it's like using hydrogen. And that's the enzyme. And then we extract the enzyme, not with these tweezers. And then basically makes light, which is not really true. We have like attached it to kind of a circuit and like in the lab and made electricity, but not anywhere near that much. And so this gained a lot of attention from just people thinking some crazy things. So I picked out some comments I thought were pretty funny. A lot of people were convinced that we would die because we've like invented something that's just too good to be true. So they thought the government was essentially going to kill us. So people thought if it was true, they'd now be dead to avoid worldwide war. Fair enough. Someone thought that the government's now going to tax us all for breathing because we're using like, you know, this finite resource that's stopping us all from having electricity. Again, this is the same thing of how we're going to like, the government's going to take our air and develop it for the masses, and so we can't really have this technology because it's, you know, exploiting the thing we need freely. We also get some crazy emails, like directly to our accounts and also to like Monash Uni. Someone sent us an email actually in Spanish. It was really weird, and we actually had someone in our lab who spoke Spanish. And it was someone emailing if we could potentially power the Canary Islands with this enzyme. And I don't really know if the Canary Islands need electricity, but they were convinced. My favorite email, though, I'm pretty sure it's from a bot, is crazy. I have like a little screenshot of it. So the subject was, I'm a mum. And the email is, Ashleigh, I've been electrocuted by my sister's home and I've had unusual circumstances happening to me thereafter. Would you know how to recognize these circumstances active in a different form? I've noticed something published about an enzyme that was published by you in a concept of clean energy and electric occurrences. I don't know what it means, but I mean, I think it's nice that people are talking about it. So, I mean, this highlights the problem of like communicating basic research, right? Like, we did something that like has cool implications, but it is basic research in that it's just exploratory. And it's really hard to convey that to the general public because it's important, because obviously taxpayers are funding our research, and so it's really critical that we tell them what we're doing. But communicating in a way that makes it make sense to them and something that they can see happening is really hard, because realistically this is not going to happen in anyone's, well, probably not my lifetime, I don't foresee. And it really requires much more investment than people think. And so the reality of the situation is the bacteria is actually called Mycobacterium sphygmatis, and this is on an agar plate. And fun fact is actually originally, it's a soil bacteria, but it actually grows on the body as well. And just like neutral, it doesn't do anything. And you can probably guess where it was isolated from, sphygmatis. It really weirdly smells like cheesy dirt, which is kind of weird. And it grows really slowly though, because it's a soil bacteria. So it takes like a week to grow. And to make it make the enzyme that we want to harvest, we have to make it starve. So that makes it grow even slower. And then this is like a very large incubator growing only 6 litres of cells. So to grow that much cells, you get 0.2 milligrams of protein, which is nothing. It's like a tiny, tiny amount. And so realistically, to do anything with it, to make any electricity or anything like that, it would require a huge time investment, huge money investment, which academia just doesn't have, and also people investment. You'd have to be growing, these things in like a bioreactor and then extracting them, on a very large scale, and then actually having the technology to see if we could actually put it on a chip to like generate some sort of electricity that's usable. So realistically, it's quite far off. But why do we do this? So this picture is just of obviously seedling and dirt. And I think a lot of people would look at this and just look at the seedling and think that's the only life there. But realistically, in the soil, there's millions of bacterial cells, and they're really critical to the planet, to planetary health, and to how everything is kind of working. So in the soil, obviously, there's bacteria that are mediating the gases in the atmosphere, because it's not just hydrogen they use. There's other gases like methane, carbon monoxide, carbon dioxide, nitrous oxide, and they're all regulating the levels in the atmosphere. And they're often the main contributors to the levels of these gases, which is hugely implicated in climate change. And then there's also the fact that, if they're using gases and they can use gases in any environment, that means that potentially they have links to the first life forms on Earth. So they're, linking matter to then creation, or kind of like the beginning of life, because they can exist without requiring organic substrates like sugars or requiring oxygen. And then that also links, I guess, to life on other planets potentially. Like planets have different gas mixes, and they have, stuff going on that is common to Earth, but not common in the way we think, water and carbon. So potentially, bacteria are living on other planets and we just don't know it because we haven't looked at things that they need, like hydrogen gas or methane. And so there's actually this really cool thing called primary succession, where basically if you have a new environment forms, so say a volcano erupts and lava flows out of the volcano and covers soil, it's completely sterile, right? There's nothing living on it. And bacteria are often the first things to occupy it because they can live off nothing. They can live off just the air surrounding that new environment. And so it's really cool to think of it in that way, that we're learning about these basic things for basic research to understand how kind of, life is formed, how life can form elsewhere, as well as potentially, creating electricity, I guess. Thanks for listening.
What an awesome talk. Didn't know that bacteria could survive on air. I'm sure we've got heaps of questions, so if we could get some lights tests. Yep, right in the middle there.
In the process of using the enzyme destroyed when it makes the electricity in the lab?
So is the enzyme destroyed when it makes electricity?
No, it just keeps going. So that's like one of the things that was like cool about this enzyme, is that it's like pretty hardy. It can just like survive again and again. And like eventually over time, like over a course of a number of days, it starts to die down. But that's usually because the products it's producing are affecting it, but not from just using hydrogen itself.
We love a renewable.
Yeah.
Yes, one in the back there.
So you said it's quite hard to actually extract the enzyme from the bacteria. Why not just take the genes, put them on a plastic collar, and mask them that way? They're okay.
Can we synthetically make this enzyme?
You can't actually, because they require, and this is the sort of thing I'm researching now, they require really extensive assembly. So they have like 10 other proteins that make them, and they have really complicated metallic centers that are critical for the enzyme to function. And so you can't actually make them in E. coli. That would be ideal. And that's kind of what we're trying to figure out now, is if we could make them in E. coli, what would be required to make them in E. coli?
Very cool. Any other questions out there? I have a question. Do we know if any other enzymes can do this? Or is it specific for this enzyme?
No, so there's heaps that can do it. So a lot of bacteria are capable of using gases, and so they all have like different forms of these sort of enzymes. And so the actual bacteria that I work on has another one that uses carbon monoxide. So it does essentially the same thing, but with carbon monoxide as the gas. And so it's actually really widespread.
Yeah, that's very cool. Yep.
You mentioned like bacteria eating the gases,
can bacteria fly through air?
Yeah, so someone from my lab is actually investigating this, that I think there is bacteria in the air. It's just very hard to study it because you have to try and extract large volumes of air and then like somehow get the bacteria into like a filter and then propagate it. And that they've been doing that at work where they just kind of have this like thing sucking air for a very long time, trying to like get some bacteria.
Maybe you just need to get everyone's like air purifiers or something like
literally.
that. Any more questions before we wrap up? Maybe last question right here. Oh, that is a very good question. How do we trust what's right and not right in the media? And I feel like COVID was the classic example, hey? I'll let Ashleigh answer that one.
I guess reading like the primary source, which is tricky because a lot of these papers are quite complex, but often the authors of the paper will so for instance, when we did this, we spoke directly to media and we were quoted directly. And then there were publications like the Daily Mail who were quoting us from a quote from a different thing, and they were obviously misquoting. And so it's like making sure you get the primary source. And so if someone specifically interviewed you or it's coming from Monash put out obviously stuff too. So reading those things rather than reading the weird Daily Mail type things.
Don't believe anything the Daily Mail says, basically.
Yes.
All right, we might wrap up there. Join me in thanking Ash again for a fabulous talk.